che devi sentire perché fai sottotitoli in tempo reale.
Prova. Prova. Uno.
È già acceso.
Capisce? Ah, ok. Ma iniziamo? Ok. Possiamo iniziare? Ok. Did they feed you well?
Yes,
Yes, good. Okay. Everything. And also the dessert, tutto qua. Ok. Buonasera a tutti. Va? Sì, grazie per essere qui. Io non voglio togliere tempo che invece può essere speso molto meglio dando tutto quello che serve al professor Hirt, Robert, che deve raccontarci insomma delle cose molto interessanti. Sono qui solo per dare il benvenuto al professor Hirt qui all'Università di Sassari, in particolare nel nostro dipartimento, con cui lui collabora in realtà con diciamo il gruppo di microbiologi, una collaborazione di lunga data che mi è stato detto in realtà è diventata un'amicizia. Ecco, quindi ho il piacere e l'onore di dare il benvenuto a questo collega, quindi amico del Dipartimento, e lascio la parola alla professoressa Rappelli che ha l'onore appunto di presentarvi questo ricercatore molto molto brillante.
Grazie Franca, io la presentazione la farò in italiano, ma non illudetevi, il seminario si terrà in inglese ma con i sottotitoli in italiano, quindi sarà sicuramente facile da seguire. Comincio col dirvi che per me è davvero un piacere, un onore poter ospitare Robert Hirt nel nostro dipartimento. Lui è professore emerito di parasitologia evolutiva alla Newcastle University. Si è laureato in biochimica, biologia molecolare e zoologia all'Università di Losanna in Svizzera nel 1987. e ha preso il PhD in biologia molecolare sempre all'Università di Losanna nel 92. Poi ha lavorato al Museo di Storia Naturale di Londra, dove ha fatto un postdoc, dove si è dedicato allo studio dell'evoluzione molecolare dei protozoi ciliati. E dal 2004, from 200, yes, lavora alla Newcastle University, dove ha sviluppato un'intensa attività di ricerca e dove ha insegnato e continua ad insegnare parassitologia, biologia molecolare, e si occupa in particolare di studiare le interazioni tra microrganismi eucarioti, l'ambiente e l'ospite. I suoi studi combinano genomica, biologia cellulare ed evolutiva in una prospettiva One Health, partendo dal presupposto che la salute umana, la salute animale e quella ambientale sono strettamente interconnesse. Robert è uno dei principali esperti dello studio dei protozoi parassiti e in particolare si è dedicato allo studio dei trichomonas e dei microsporidi, che saranno l'oggetto dei 2 seminari che terrà oggi qui da noi. Questi sono studiati come modelli che permettono una maggiore comprensione dell'interazione tra i parassiti e l'ospite, attraverso lo studio dei meccanismi molecolari che regolano proprio questa interazione. E in particolare ha studiato anche l'interazione con i microbiota, sia a livello intestinale, vaginale che respiratorio. Come diceva già la nostra direttrice, la collaborazione con Robert Hirt è iniziata più di 20 anni fa, quando lui ha partecipato a una international conference sui protozoi a Nairobi, che si è tenuta ad Alghero, e da quel momento abbiamo sempre collaborato e questa collaborazione è stata anche rafforzata dal soggiorno di Valentina Margherita, che allora era una dottoranda e che adesso è una ricercatrice del nostro dipartimento, che ha contribuito a mantenere questo ponte tra Newcastle e Sassari. In realtà, come diceva Franca, Robert non è solo un collega, è più di un collega, è un amico, e questa collaborazione ha permesso di sviluppare tante idee, ma anche tanto divertimento, e quindi devo dire che è con grande piacere che invito Robert a presentare i 2 seminari, che spero saranno un'occasione per alcuni di approfondire argomenti magari già conosciuti, visti sotto nuovi punti di vista, e per i più giovani saranno anche l'occasione di vedere cosa c'è dietro quello che studiate, come si arriva alle certezze che voi trovate sui libri attraverso lo studio e attraverso la ricerca. Il seminario sarà diviso in 2 parti e la prima parte è intitolata Microsporidial Epithelial Cells Interactions, What Role for Bacterial Secondary Metabolites. Quindi do la parola al professor Hirt e prego. Ho parlato in italiano perché lui lo capisce da una parte e perché quello che ho detto me l'ha detto lui e quindi...
Unfortunately, I don't speak Italian. I don't understand some of it, but although in Switzerland, quite a few people do speak Italian, but I'm from the French part, I'm afraid. So I will stick to probably English is probably better. So thank you for the introduction, and thank you very much for re-inviting me here, because it's not the first time I've been coming here, and it's really wonderful to be back. Full screen.
I'm trying to do that.
Yeah, that should work. Yeah. Thank you. Thank you very much. Hooray. We are set. Is there a laser by any chance? So I forgot to ask about that. Input? No, I don't touch that. Is there? I can just use that. No, to move, but to show the screen. Anyway, let's start. So I just wanted to first thank you, Gigi and Paola and Valentina, for the wonderful hospitality. And it's not only scientific hospitality, but also at the level of, as Paola said, we became friends. And also Alina, my wife, is not here, but we thoroughly enjoyed coming as a couple, as we are both scientists as well. And it was wonderful to be here to meet. The first person we met actually was Daniele. Where is Daniele? Is he in the room? Desi. He's not here, okay. And he was, sorry? Okay, thank you. Anyway, it was just, and that from that moment, we kind of fell in love scientifically and also enjoyed discovering Italian lifestyle and Sardinian lifestyle in particular on the food. So there's many things we enjoyed coming here, and in particular this famous meeting in Alghero. That was sort of the start of many things that we did afterwards. So I will talk about some of those data. More generally speaking, what I'm going to talk about is my interest in trying to understand how parasites interact with the host and how is that influenced by the member of the microbiota, and how also other aspects of the environment, in particular the diet, can modulate those activities. That's going to be relevant in particular when we talk about Microsporia. So we're going to have two very distinct kind of parasites, Microsporia strict intracellular parasites, and then Trichomonas extracellular parasites. And we're going to see the kind of interaction they have with the host and how those interaction, thank you very much, can be modulated by the bacteria. So, and also we're going to mention a little bit about viruses as well, okay? So that's why I like this kind of scheme, which is sort of the new paradigm to understand host-microbe interaction, where we need to integrate many factors. We need to look at the eukaryotes. That's one of our main interest from Paola, Valentina, Daniele, and so on, the trichomonads in particular, and how those interactions are very much dependent on many factors, including what bacteria are around. And what hopefully I'll be able to convince you by the end of the presentation is the importance to study that, not just if you're interested in humans, but also to look at the animal hosts, from where many of these organisms come from. And in particular, hopefully I will convince you today that if you're interested in trichomonas, you've got to work on pigeons, okay? So we'll see about that. We'll see if I'm able to convince you that way. So 3 parts. First, the general introduction, and then what's the relevance of microbes at mucosal surfaces, a little bit of semantics, and then we're going to have the first part, as Paola said, we're going to talk about the Microsporia epithelial cell interaction, and we're going to talk about, in particular, short-chain fatty acid. The second part, we're going to talk about trichomonads, which is one of the main reasons I came here initially, was to work on why we collaborated over many years. Okay, so a bit of semantics first. I like to use the word symbiosis as it was actually defined by Dubarry. Okay, Anton Dubarry. It's a French name, but actually the guy was in Germany. I was told by one of his ancestors that I happened to meet by chance that they're all from Belgium somewhere. Okay, so it was very funny because he's from Swiss German part. I speak French. I have a German name. He has a French name. He speaks German. Anyway, just mix up of everything. Very interesting. Anyway, so this is the person who defined symbiosis, and he defined it as the association between organisms from different species, and there are 3 major categories of interactions. Mutualistic one, everybody's happy, everybody benefits from those interactions. Commensalism, one partner benefits, the other one doesn't do any much. And unfortunately, these words are used in slightly different ways by different people, which confuses a bit things. That's why I thought I start with the semantic bit. A lot of people now mention commensalism when in fact they want to say mutualist. Okay, so I thought we'll talk about that. Then parasitism is one partner is desperate for a host, and when you interact with this host, it can cause damage. But as we're going to see, the damage, if there is any damage, is very much context dependent. And that's one of the things that we're going to look at, how the microbiota gets involved. Okay, among other things as well. So here, just to make, I'm going to zoom actually right to the bottom part here because I've talking too much already, and we had lots of introductions. The main thing I really want you to take from here is the outcome of host-parasite interaction depends on the context of those interactions, and very important, that depends on not only on the host, but also the environment, and in particular microbiota. A human being or any animal is not just human cells and genomes, it's a combination of human cells and genomes, tissues and so on, and whatever organism they're interacting with. Probably the best way to think about that is we're on a planet that is microbial, essentially. Okay? And as microbes have been around for probably more than 3 billion years ago. We came much later. So everything on this planet works with microorganisms. So if we forget that, we kind of forget an important aspect of reality of our world. And that includes our own phenotype, our own functionalities, okay? So hopefully at the end of this, I will illustrate that in a hopefully convincing way, an interesting way for you to think each time you think about human well-being, human disease, or animal well-being, animal disease. I've got those lectures covered. Hopefully that will be of interest for some of the medical students, but also hopefully of interest for the biologist, okay? So I have this kind of dual interest. Hopefully that would work. And one thing that maybe we should say, because parasites don't always cause disease, maybe we should talk about them as pathobionts. They cause disease in some context. ¿Qué pasó? No? Bueno. Okay, let's keep going. So, one of the most interesting places to look at host microorganism interaction is at mucosal surface, okay? And it's interesting because it's very complicated, but also central to almost anything, okay? If the mucosa doesn't work, you're not going to breathe. If the mucosa doesn't work, you're not going to be able to digest and absorb nutrients, and you won't be able to reproduce. All of those features I just mentioned are all dependent on appropriate host tissues, cells, genes, etc. But they also depend on the microbiota. And what is very interesting and very intriguing is that the mucosal surfaces need to do two very contradictory things at the same time. So I think that's an important thing to think about. And these two activities are dependent on relationship with the microbes. So you only get an appropriate protection at the mucosa if you have an appropriate microbiota as well associated with that mucosa. And then the exchange between the outside and the inside of the body is also going to be optimal only if you get an optimal microbiota. So these two contradictory functions forming a barrier on one side to avoid infection, to avoid damage, and at the same time facilitate exchange, essential for breathing, essential for nutrient absorption, and essential for reproduction. None of that works properly if we don't have proper interaction with the microorganisms. So that's what I like to illustrate here. We have complex interaction between microbial eukaryotes, bacteria. In the gut, for example, most of the biomass would be this, and there will be a few microbial eukaryotes. But in some contexts, as we're going to see, the portion of these bacteria, microbial eukaryote can change and can become condition what leads to pathologies, for example. And then we've got the viruses as well. So we're going to talk a little bit about viruses when we're going to talk about Trichomonas later on. But the point I want to make here is that we have a very intense, continuous interaction between the microbes and with the host, with the mucosa. And the mucosa only is going to be able to do its job, protecting and absorbing, facilitating exchange and so on, if everybody works or dance in the same tune, basically. Okay? So they will modulate activity of the epithelial cells, they will modulate immunocytes. So you have this very complex set of interaction between the microbes, epithelial cells, and immunocytes that are required for that mucosa to do its job. Okay? Now, what is interesting now with over the years, we understand that you can actually have disease by simply shifting the proportion of different organisms, different activities, even without specific pathogens. There's lots of condition, IBD and so on. You don't necessarily need a pathogen. And that leads to the concept of dysbiosis, where that means it's a combination of microorganism activities that can cause disease, okay? And that contrasts with eubiotic microbiota, which promote health, okay? And there is no health without these microbes. So it's not a question whether, microbes bad and so on. No, we need those guys. What is important is to have them functioning in the right way. And this is where that would also depend on what we eat, for example. Okay? So depending what we eat will modulate what's going on here as well. So the key concept here is that you can shift from a state of homeostasis to a disease state, okay, if you get the wrong combination of activities of microbes, and these activity of microbes going to be dependent on who is there, what environmental factors modulate them. Smoking, for example, will modulate activities of the microbiota in your mouth, in the gut. And then your diet, we're going to see the diet, very important for short-chain fatty acids. And then the host. The host must be able to interact in an appropriate way. There must be enough mucus, enough antimicrobial peptide, enough antibodies, etc. Okay? And all of that is required for this amazingly complex system to work. Okay? So here's a kind of nice little review that illustrates the importance of microbes. And here they use the terms in the wrong way, I think. They say symbionts for when they say mutualist, and they use commensals. Well, maybe they're commensals, that's probably all right. And they talk about parasite pathogen on this side. So you need an appropriate balancing activities of these different microbes to maintain homeostasis, okay? And here are some of the key factors that can cause problems and lead to shifts. That can be host genetics, if you have some genetic background that make you more susceptible, depending on the diet, depending how you were born, for example, can impact what kind of microbes you have. And then if you use medication, all of those factors can directly or indirectly lead to dysbiosis, which can then potentially promote certain diseases. So where does the parasite sit in there? So parasites, or the ones that we typically call parasites, it's a bit more complicated than that because some of them might be actually mutualistic in some context. Okay? For example, Trichomonas muris in the mouse can make a mouse more resistant to salmonella, for example. Okay? So it's a bit more complicated than that. But in the context of what we've worked on with Gigi and Paola and Valentina is there's clear evidence that organisms such as Trichomonas are associated with this very different combination of microbes that are more disease-promoting than actually health-maintaining. And the things I've been interested in is to understand the molecular basis of those interactions on using in vitro system. We're going to talk about that with Microsporia, for example, and also culturing system where we use comparative genomics, comparative transcriptomics, and some functional assay to better understand how those organisms interact with each other and how that might promote or cause disease. Okay, so we're going to have these three parts. First, an introduction, and we're going to simply talk a little bit about gene sharing between them. And then in the first part, we'll talk about data. I'm going to talk about Microsporia and how interaction between epithelial cells and Microsporia can be modulated by short-chain fatty acid produced by the microbiota. Okay? In the second part, after that, I think we should have a break, right? Yeah. In the middle. So after the break, we can then move into Trichomonas, which is why I came here. Okay? Okay, this is just to remind you, Microsporia, I mentioned that earlier. Microsporia are strict intercellular parasites. They have to go in the host cell. If they can't go in, they can't do anything. It's like a virus or rickettsia or chlamydia, okay? What is fascinating, they have to do that coming in without killing the host cell. Because if they kill the host cell, they are done, okay? And we're going to see they do that in a very peculiar and amazing way. They use a tube made of protein to do that, okay? And then for Trichomonas, these are extracellular, and they are highly adapted, highly sophisticated organisms that are able to interact with mucosal surfaces and the microbiota. And that's one of the things that'd be really interesting is what's the molecular basis of the interaction. And that's where we share a lot of interest with Gigi and Paola. In terms of genomes, these two kind of parasites are completely the opposite. Trichomonads are some of the most complex genome in terms of coding capacity. That's an old number. The latest number, up to date with a complete genome where they sequenced everything from A to Z, they have more than 77,000 genes, protein coding genes. They're not all functional, but there's lots of them. In the opposite end, Microsporia, I'm going to talk about these two a little bit. They have less than 2,000 protein-coding genes, so half of what E. coli has. Okay? So these are all microbial eukaryotes. They're all parasitic. They can cause disease. They can take advantage of the host. They do that in very different ways. Okay? And that's reflected in very different genome sizes and coding capacity. Okay, so first a little bit about one of the things I got interested when we looked at parasites. Where do they get genes? And one of the things that were discussed at the time is any relevance for lateral gene transfer. We know that's really important for bacteria. It's the main source of new genes between bacteria. What about parasitic protozoa? So we did this study here, and I'm just going to mention a few things. Trichomonas was one of the champions. We used, for this, these numbers are very conservative. We use very strict rules to select genes for which there's enough phylogenetic information. And for Trichomonas, we had more than 100 genes with very clear evidence for lateral gene transfer. I will show you some example of that. And as we move forward in the second part, the second talk after the break, we're going to illustrate some of the importance of those genes in how Trichomonas interact with the microbiota. Okay? So here's an example of a gene that you find in Trichomonas vaginalis. We're going to talk that back again, its functional relevance, where it's clearly coming from Bacteroides. Bacteroides, where do we find Bacteroides? I understand you're going to have questions after that. You have to answer, right? So I'm asking you a question. Where do you find Bacteroides mostly? Come on. You never heard of Bacteroides? Por favor, Bacteroides. So these are bacteria that we found in the colon, mostly. Okay? So it's one of the most abundant and common bacteria in the colon. So it's interesting that Trichomonas has shared gene, and it's very similar to the protein. If you do a blast, the protein are extremely similar. This is an interesting gene. It's involved in it's in a pool, polysaccharide uridinylose, that Bacteroides use to do what? To consume mucus. So we have a nice paper with that. I'm not going to talk about this, but I'm going to talk what that does in Trichomonas and why it's relevant, potentially in the context of interaction with bacteria. Okay? So here a clear case. The gene is transcribed. It's functional. We know all of that. So it's not a contamination. So that's the kind of example we have. Very few trees look like this. This is sort of tree of life. You have eukaryotes, archaea, and then bacteria. If you zoom here, you have again Trichomonas, and bingo, again, Bacteroides. So there's quite a few of those. And if we look at the overall stats, you can see there's a very biased distribution across different bacterial lineages, with most of the mucosal one having a strong bias for Bacteroides, which are found commonly in mucosa. So basically, you live in the same place, you kind of share genes with each other. Okay? In the same way we shared dinners together, right? So they like each other. Okay, what do these genes do? They're mostly enzymes. Amino acid metabolism, quite common. Carbohydrate metabolism, we're going to talk about that when we talk about how Trichomonas interact with bacteria. And then I would like to also highlight the fact a big fraction of those genes, we've got no idea what they do. So that's really interesting. There are genes shared between bacteria and protozoa. The protozoa keep them and do something with it. We don't know what it is. But they must be doing something important. Otherwise, they would not be going around and not be conserved. Okay? So that's an interesting question for future research projects. Okay, so just a summary. We've got lots of LGTs. And as I said, we're going to talk about this later on in the second part, where we're going to see some really interesting, well, we find them interesting, how Trichomonas interacts and mediates specific interaction, in particular with the cell wall of bacteria, for example. Okay? All right, so let's talk about Microsporia now. Okay? So these are the intracellular parasites. I'm going to give a brief introduction. I don't know if you had any lectures on Microsporia. No? Okay, here we go. So I'll try to do my best for you to remember all of this. So there are strict intracellular parasites, okay? As I said, they're like viruses, rickettsia, chlamydia. They have to go inside the host cell. What is very peculiar and unique and fascinating They do that using something called a polar tube. It's a tube made of protein, complex of proteins, that is coiled within the spore. And if you do a section with transmission electron microscopy, you can see that each of those little dots here is a section across that tube. So it's coiled within the spore, and then it gets extruded on the end here, and then it's used to somehow, we'll talk about that later in more detail, to go inside the host cell. Once inside, it takes control of it like a virus, consumes it, and makes more of itself, okay? So we have been interested in understanding how they can get nutrients and energy from the host, and then we're going to see how that also modulates a key function of the mammalian cell, autophagy, okay? They infect animals, very common among insects. If you like to eat shrimps and so on, well, they cause problems in farms. This is actually a very similar species to one that is more common among humans if you have HIV/AIDS. Very closely related to these guys here. They infect humans, and that really showed up on the radar of medics when there was HIV/AIDS. So when HIV/AIDS came up, people didn't understand what's going on initially, then discovered the virus. But then there was a whole range of diseases that turned up that people were not very familiar. One of them was microsporidia, okay? So if you have the CD4 cells that crashes down to a level, microsporia can start to cause a lot of problems, okay? And in particular, in the gut. So we're going to talk about that in the context of species that infect typically the gut. How do microsporia infect? As I told you, they use a tube. It's not clear exactly how they do it. At the time, here's a review with a nice cartoon. That's the yellow bit. That's the polar tube. It's made of protein. It's expelled, and it can perforate the membrane. So there's clear data showing that for some species. And somehow the parasite is pushed through that tube, okay, and end up on the other side. There's also evidence that the microsporia spore can sit on the surface of epithelial cells and get internalized, and then use the tube to come out of it, of that membrane, okay? There is some nice data from French and US people and Chinese collaborators with them as well that shows that for encephalozoon, cuniculi, intestinalis, and so on, it could be that the tubes comes out, interact in a glycan-specific way, and then gets invaginated, and the tube deposits the parasite in that vesicle that then becomes internalized in an actin-dependent way, okay? So it's a unique system. Nobody else does that. That's the microsporidia cool way of going in, and remember, without killing the host cell. If you kill the host cell, then there's nothing you can actually exploit, okay? So we've been interested in understanding how that once they're inside, what do they do? First, they don't do much because they have lost many genes, and many metabolic pathways are really skeletal. There's not much left, so I'll just highlight some of the things here. I'm only going to talk about 2 transporters, and this is one of the few things where we look at the numbers of genes, where the genes either were acquired, so they've lost many, many genes, right? Many metabolic enzymes, but they have acquired a few things. That includes the NTT transporter, and we're going to see that these ones are involved in stealing nucleotides and energy, okay? So they don't bother doing ATP and so on, except in the spore a little bit, but they actually take it directly from the host. So we're going to look at how that works. So we started this by looking, at the time there was a genome of encephalozoon cuniculi. We looked at the genome, we found those transporters that were discussed by the paper, and we couldn't resist to try to work on that. So we have a fantastic PhD student, Tassos, from Cyprus, and we made little peptides. We made a kind of crazy combination of peptides together, injected that into to get rabbit antibodies, and we could demonstrate, as we would expect, that these things are on the surface. Why do you expect that? Because rickettsia and chlamydia do the same thing. Okay, so when chlamydia and rickettsia infect you, they are intracellular, and they will steal your energy directly from your host cells, from your cells. So we could demonstrate they can transport nucleotide. We had to do that in E. coli. Okay, so it's a nightmare to do, I can tell you. He worked a lot. Then Eva expanded this, and we tested that in another microsporia. I'm not going to go into detail, but basically we can show that they are nicely expressed on the surface during the infection cycle. So really essential transporter, and they can transport pyrimidines, okay? Purines. Then Paul Dean, another postdoc, went into the detail of how the transport works. So this kind of transporter can be exchangers, or they can be symporters. You met Paul, no? Yeah. And so what Paul could demonstrate is they both use symporters and symporters and exchanger. And he also could demonstrate, because we did ancestral sequence reconstruction, that the first transporter was likely an ATP-ADP exchanger, just to steal energy. That's the only thing he was involved with, the ancestor state. I'm not going to say much more than that, except that now we know that they have symporters. That's for net import of ATP, GTP on NAD as well. They have exchangers to keep balancing the oxidoredox balancing, and they use exchangers to steal ATP on GDP. So they have this machinery to be able to bring in everything they need for their own energy, okay? There's one thing we could not find out, is how they get the pyrimidines. None of those transporters touch pyrimidines. In chlamydia and rickettsia, they have transporters that do everything. Pyrimidines, purines, the lot, okay? Not in microsporia. There's no data that we could figure out. There's no evidence that they could bring in pyrimidines. So we got very excited when we thought, oh, let's try these ones. So that was Peter Major, who was a postdoc in the lab as well, worked with Casper and Paul Dean in particular. Same thing, we'll expect them on the cell surface. They were, bingo, that was great. And then we said, okay, let's have a look what they transport using the same assay for the entities. Great result, but no pyrimidines either. So they can also import GTP on ATP. They are all exchangers. So it looks like they use two sets of transporters for doing the same kind of job to steal energy. So we still don't know how they do the pyrimidine. So maybe some of you will do a project that will determine how the microsporia get the pyrimidines. We don't know. We have no clue. So the next thing we're interested in to look at, that was with Johan Panek, a postdoc from France. He did a very nice postdoc in the US with someone who works at UCSD on C. elegans model to study microsporia interaction. And they had some interesting data suggesting that autophagy, or more specifically xenophagy, was controlling infection in C. elegans of that. So we had talked about before you went to the US, maybe we should do the same thing in the mammals, because we don't know how the microsporia interact. We know how they can steal energy. And if something steals energy, the host cell will be stressed. You need to respond to that. And one way to respond to this is through autophagy. Okay? So we were predicting that autophagy is probably something going to be relevant not in C. elegans, but also in mammals. So Johan set up a nice project. He got a Marie Curie to do that. And he was unfortunately disrupted by COVID. It was a nightmare. But we eventually got some nice data and got this paper published. And so these are the questions that he set up to answer. And that included in particular the use, we tested short-chain fatty acids. Now, had you had any lectures on short-chain fatty acids? Yes, no, sí, no, sí, no, no. In Argentina, it's very funny. Sometimes there's a conversation, and then they say, sí, no, no. And at the end, what did they say? Yes or no? So, what is the answer? Have you had lectures on short-chain fatty acids? See? No, see? Well, read about it, because it's very important, and I hopefully will convince you that it is. So, the idea was he looked at two kind of measurements. One is how many spores are produced. That's the final stage of the infection. Okay, and then he also looked at the size of the structures in which the merons divide and will eventually make spores. Okay, so he looked at how many spores are produced and what's the size of the vesicles that contain the parasite when they are growing. Okay, after 48 hours. So he looked at how drugs that modulate the activity of autophagy will modulate this. And he also used genetics by downregulating a key factor of autophagy, ATG5. So if you knock out that gene, autophagy is stopped. Okay, so what happens when we do that? And then short-chain fatty acid can be, it depends the context. Some cancer cells with short fatty acid autophagy is stimulated, and in healthy cells, normally epithelium, short fatty acid inhibit autophagy. So that's context dependent. So if you're interested in cancer biology, be aware of that. Now, so what happened is he tested 2 drugs that are known to increase autophagy. So one of the idea was, if you increase autophagy and you happen to stimulate xenophagy, you're going to destroy the parasite. Okay? Did we see that? No. In the contrary, it was stimulating. So here we look at the size of the parasitivorous vacuole, the vacuole where the parasite is growing. And the more parasite there is, the bigger the vesicule becomes. Okay? And you can see that as you increase the dose of taurine or the dose of rapamycin, you typically get an increase rather than a decrease. Okay? And these are the numbers here for the numbers of spores. So instead of destroying the parasite through xenophagy, you actually stimulate more spore production. Okay? What about inhibiting? So if you use a scrambled sRNA that is not targeting anything specific, it's the same composition as the specific small interfering RNA that targets ATG5, you can see that nothing happens. That's as you would expect with control. You have certain numbers of a certain size of parasitic vacuole. If you use taurine, it goes up, as we shown earlier. Now, what happens if you do the sRNA that target ATG5? That means you're going to inhibit autophagy. You have a dramatic reduction in numbers of the size of the vesicles. Okay, so it looks like there's a link between how much autophagy is happening and how happy the parasite is. Okay, quite interesting. So the idea is that autophagy is this system that allows the cell to recycle protein complex, organelles, peroxisome, mitochondria. So this can be all recycled, and then when you have autophagy fusing with lysosomes, you're going to destroy everything in that vesicle and release components that can then be used by the cell to stay alive. Okay, so that's switched off, for example, if you don't have any amino acids or you don't have enough energy. Okay, so it allows to recycle some component to try to keep the cell alive. Now, if autophagy was targeting the parasite, then you would expect the parasite to be going into those vesicles and get destroyed. And we know that can happen, for example, with salmonella. Okay, so salmonella would be destroyed. Or some parasites like malaria in some tissue get destroyed that way. Okay, so we were speculating that maybe something similar could happen, but clearly we saw the opposite. Despite the fact that the microsporia was labeled with ubiquitin. So we could see that. But that was not enough. For some reason, somehow, and we have no idea how that works, the parasite is able to avoid xenophagy. So instead of saying xenophagy, where you will have prediction destruction of the parasite, we saw the opposite, where autophagy was actually promoting, and what we think is happening is that you get transfer of biomass from the host cell to the parasite. Okay? So I think I've got, yes. So what I'm illustrating here is the cycle. Remember, you go in, you're going to steal energy, you're going to steal amino acids, and the first thing, one of the first things the mammalian cell will do, that will switch on autophagy. Okay? And in principle, it will also try to stimulate xenophagy, but that is clearly not happening. And then it progresses and then makes more spores of itself, and then the cell will lyse. So what seems to happen is that autophagy is clearly stimulated. You have recycling of the organelle, mitochondria, peroxisome, protein complexes, then fusion with the lysosome, okay? And that liberates those components that are degraded from the organelle. This component will be basically sucked up. It's like a Hoover by the parasite, okay? So that will allow to transfer, we think the hypothesis that autophagy, by consuming the host cell biomass, then is able to consume, to hoover that. I will take the hoover, and takes everything away, okay? And then the parasite is very happy, has all the constituent that it needs. So that's probably what is happening. That's our preferred hypothesis. Now, what about short-chain fatty acid? Now, if you had a lecture, very briefly, short-chain fatty acid are produced by bacteria, mostly in the gut. Okay? In the colon, you have maybe 1.5 kilogram of bacteria. Fecal material is 60%, the mass of fecal material, 60% bacteria cells. It's a lot, okay? So, short fatty acids are produced by fermentation of fiber. That's why you need to eat fiber. And that's why the Mediterranean diet is very good, because it's rich in, well, it's rich in many good things, and I love the food. So, yes, keep eating Mediterranean diet if you do. One of the important things is the fiber. So the higher your fiber in the diet, in combination, that's the key thing. You can have as many fiber you want. If you don't have the right bacteria to digest them, you're not going to have the right proportion of short fatty acids. Now, short fatty acids, why are they important here? They are central to the gut homeostasis. It will determine how much mucus your gut produces. It will determine how many Paneth cells will produce antimicrobial peptide. It will determine how healthy those cells are, because it's the main source of energy of your colonocytes. It's not your diet. It's your diet via the bacteria that make the short fatty acid, and then that feeds your colon. Okay? So really central. So if that is not happening in an appropriate amount, your gut is going to be suboptimal. Okay? Another interesting thing is, because the colonic cells are happy with the short chain fatty acid as a source of energy, that will typically shut down and reduce autophagy. Okay? So we were interested in checking that. So Johan checked that in this system on epithelial cells grown on plastic. I will now show later the result when we grow colonocytes, CACO2 cells, on filters, where you can reproduce to some extent the physiological context that epithelial cells get nutrients from both sides, in particular the basal side, because there are pores here. When you grow them on plastic, you don't have that. They only get access to nutrients from the top, which is not really what happens in the gut. Okay? But so we can then look at what happens to tight junctions. There's a link between autophagy and tight junctions. The autophagy goes up, the tight junction gets a bit nibbled too. They can get destroyed. And then if you have a good amount of short-chain fatty acid, you're going to have reduction of autophagy and perfectly functional, happy little colonocytes. So what happens if you use short fatty acids? You actually reduce significantly spore production. Okay? So that will just be your diet. Depending what you eat, you might be more or less susceptible. Well, eat and composition of the microbiota, you might become more or less susceptible to infection by Mycosporia, even if you are immunocompetent. Something of interest to think about. Okay, so next I'm going to show you some data from Moody bin Saleh. She's a PhD student from Saudi Arabia. She's currently writing up. And we basically, the key aspect of the project was to expand those interesting data and see what happens in the barrier function, not just how much spores were produced and so on, but also the barrier function, how good the epithelium is to separate the outside from the inside, okay? Which is a key function of the gut lining, okay? Which can go wrong, for example, in quite a few conditions such as inflammatory bowel disease. So what happens is the following. So she grows a cell, CACO2 cells, about 2 weeks, and then 2 weeks you get a nice monolayer, and you can measure transepithelial resistance. It's how tight it is. You can get high level. That's after 2 weeks of growing those colonocytes. It's a standard protocol. A lot of people use CACO2 cells, and I've seen some poster with CACO2 cells here. So here we go. Now, what happens if you have nothing? That's the control. That's how, for 6 days, you have a nice, stable monolayer, okay? Now, if you add spores from Ancylophyllum cuniculi, you can see the resistance going down. That's exactly what you expect. So you have an infection, it damages the cell, it consumes the cell, some cell will die, it disrupts probably the tight junctions, and therefore the transepithelial resistance go down. what happens if you add short-chain fatty acid after 2 days of infection? You get an amazing reversal. It's spectacular. I mean, I couldn't believe that. I said, what? Are you sure you had the result right? I did ask her, you know. And she reproduced that several times now. So now we've got this amazing recovery that even goes beyond the control. Okay? So it's a really interesting phenomenon. Despite infection, so somehow the damage caused here gets reversed to some extent with short fatty acid. Of course, it's in vitro, okay? In a cell line. So I'm not saying that is directly telling us what's happening in the gut, but it's interesting. That's for sure. She also looked at using beads. So you put fluorescent beads on one side, and you see to what extent they go on the other side. Okay? If you have a tight, nice tight monolayer, you will have very little flux on the other side. If you have an infection, that goes up, consistent with the reduction of the transfer resistance. And then if you have short fatty acid, you go back to control levels. Okay? So again, it kind of repairs the monolayer to some extent. Okay? Now, what happens if you look at cell viability? So that's the control. That's the control with. I always get confused. Yeah, the short fatty acid. That's just on its own. So you can see that short fatty acid actually the cells really like it. It improves the viability in that system. If you have infection, that has an impact. But if you have infection and then short fatty acid that you add either after or before the infection, it's even more protective, as you would expect. Okay? So really nice phenomenon that illustrates the importance of short fatty acid in that in vitro system, consistent with what we know is happening in vivo. Okay? So really interesting context here. And if you look at numbers of spore produced in those conditions, so that's the total numbers of spores, and that's the proportion of spores that reach the basal side, the other side. So remember, we put the spores on the apical side, right? So if the monolayer stays tight, or if there's no infection, there's no spores, then there would be very little on the other side. You can see that if you add short fatty acid, you reduce dramatically the numbers of spores produced by that in that infection model system. Okay? So really dramatic impact. So reduce infection, you possibly inhibit the parasite in one way or another, and you definitely help the epithelium to repair itself from the damage caused by the infection. Does that work in the same way for all species? Again, that's just we wanted to check another species. This one is more common in humans. So if you have HIV/AIDS, you will see more of that than this one. It can also exist. Cuniculi is because what kind of animal would be infected by something called cuniculi? Any idea? Anyone? So far, I have got 0 answers from you. It's very bad. Cuniculi refers to rabbit. So it's a common infection in rabbits. The poor rabbits, they get infection in the gut, and then it goes systemic, and then they get neurological pathologies. They kind of have their necks broke, kind of funny. It's kind of very common. And there are examples of zoonotic transmission between pets and their owners. There's a case with the guinea pigs that were infected by cuniculi, and then the family got infected. Okay, so what happens? Cuniculi, I showed you that earlier. So that's the data I show you here. You can see the impact of the infection, the magnificent recovery induced by short fatty acid. And then it's even better when you, if you do pre-incubation, you can see that the monolayer is really healthy and can cope with infection, basically. Okay, it's a bit less effective for intestinalis. It's basically a bit more virulent in a sense. So the impact is not as dramatic, using the same numbers of spores to infect, but you can see the same phenomenon essentially observed in both cases. But as I said, it's not as effective for this species, so suggesting it's more infectious. Numbers of spores, very similar picture here. Again, in both cases, you can see that short fatty acid have dramatic impact on spore production. Okay, so it's interesting. You allow the epithelium to repair itself, forming a monolayer, and you also somehow inhibit the parasite, the two things. So potentially quite interesting if that is relevant and we can reproduce that in vivo. How do they impact the tight junctions? So if we look at infection with, so the control is just the CACO2 cells on their own. This is with tight junctions with short fatty acids, and this is the infection without short fatty acid. And you can see that there's a dramatic shift upwards in terms of how much ZO1. ZO1 is a key component, I will show you in the next slide, of the tight junction. The tight junction, it does exactly what it says on the tin, which is to keep cells tight to each other, okay? So that's shown here. The tight junctions are a key aspect of any epithelium, and in the gut in particular, and allows to connect robustly the cells with each other, which ensures that there's nothing that can freely diffuse through the epithelium, okay? And the ZO1 protein is one of the protein that makes up the tight junction itself. And you can see that by immunofluorescence, that's the control, you have very nice quantity of ZO1 protein that form the tight junction itself. If you have an infection, that has a dramatic impact. So the amount of protein goes down, and that is directly correlated with the change of tear that we observe during infection, okay? And if you put short fatty acid, there's a little bit of improvement. So consistent with the data we had earlier. In terms of repair, Moody also checked if you do a very simple wound scratch test. Here's a CACO2 cell without infection. With infection, you can see that the infection delays the repair because the cells are busy feeding the parasite, okay? If you put short fatty acid, you recover to some extent the initial phenotype. So short fatty acid clearly helps the cells to repair the tissue. So really important metabolite. Remember that comes from the microbiota. So the properties of these cells that we looked at and how they respond to the challenge from the parasite is entirely modulated here by secondary metabolite from the microbiota. So that's the conclusion from this part. What's the time? Yeah, it's still okay? So just to conclude, so we show that autophagy, it's not xenophagy happening. It's actually promote the proliferation of the microsporia. Short fatty acid can inhibit both autophagy and the production of parasites. So very interesting. It could be that short fatty is actually toxic for the parasite in one way or another. We don't know. It also short fatty acid also significantly strengthens epithelial barrier integrity in the system. Wound scratch also show the same. And so depending on the diet, that's a really important thing here. Depending on which organism you have in your gut and which diet you have, you're going to have different proportion of short fatty acids, and therefore going to modulate how good your intestinal mucosa surface is protected or not. Will make you more or less susceptible to infection, such as microsporia, but it's relevant in other contexts as well, of course. Okay? Now, anyone who study or work in the gastrointestinal unit will know a big proportion of patients with gastrointestinal symptoms, 50% of the time, no idea, okay? And then it can be quite difficult to come with a specific diagnostic, okay? On microsporia, beyond the HIV/AIDS, if you have microsporia diagnosed, normally a good doctor, the first thing he does, you got an HIV test done immediately, okay? So that's well established. But there are lots of other conditions which are not obvious. One of which is all the different symptomology associated with inflammatory bowel disease, like such as Crohn's disease, ulcerative colitis, and that can be quite hard work for diagnostics. So if any of you are going to work on gastroenterology, and you're going to have some course on that, I'm sure, but anyone who's become a specialist in gastroenterology, it's an enormous challenge to come up with ways to stratify, understand IBD patient. And they typically have very heterogeneous way to respond to treatment as well. What is interesting, there is some literature suggesting that microsporia might be relevant in pediatric patients. And also here, for example, 30% in one, it's a small study in Spain, small numbers of patients, but 30% were positive for microsporia. Now, why would that happen? It could be that treatment typically is anti-inflammatory. So you actually inhibit the immune system. You try to dampen down inflammatory response. and that may make you more susceptible to infection because of that. Okay. For example, if you have anti-TNF, is it TNF alpha? I think. So I have actually a niece who had that treatment with monoclonal antibodies, and if you use that, you become more susceptible to a range of infection, including these guys here. So, final sort of take-home message: immunosuppression could potentially make you more susceptible. That can lead to biodysfunction on microbiota alteration. And to go back to the short-chain fatty acid, in IBD, typically the biomass of the microbiota goes down, and the production of short fatty acid goes also down. So here we go. You're going to have issues like that. So it could be that considering microsporia, I'm not saying that the most important thing, I'm not saying that, but it could be that it might be an important thing to consider to differentiate different kind of patients. Okay? Patients stratification, which is a key word for most patients, but particular IBD. And I think I will stop here. Yes, for the first part.
Thank you so much. I understood probably half. So, good. So, I have a question, a curiosity. My question could be trivial because I'm just, a simple neurophysiologist. So, this question is quite oriented. Okay, this is my question. So, you may know that concerning the taste sensitivity, people are divided in three main phenotypes: non-taster, bad tasters, medium tasters, and super tasters. And this distinction relies mainly on the taste of bitter taste. Yes. Yeah. And the reason is mainly genetic. So we have a genus that in some way influence this phenotype. Yeah. But the cause is not only genetic, it is also rely on the interaction of the subject with the environment, and very likely also with the interaction with these guys. Yeah, so I was just wondering if, I don't know anything about this, this topic, but I was just wondering if, the oral microbiota and the interaction of the subject, probably also early in life. Yes. So previous interaction with these guys you talked about may have an influence on this attitude, because it's quite important, because then if you are a super taster, you probably are more likely to develop, obesity or other problems, health problems. So I was just wondering if there is a relationship between this.
So I have no idea about the specific case that you mentioned, but as far as I know, there's a lot of studies where they look at the gut-brain relationship. Yes. So it could be that some aspect of that might be modulated. So your neurological capabilities in terms of tasting might potentially be modulated indirectly, but what happens in the gut, for example, or the oral cavity, as you mentioned.
We have these receptors not only in our tongue, but also in our gut. Okay. So maybe taste receptors are also.
I mean, I know there's quite a bit of studies where people contrasted the microbiota of obese people and non-obese, and then diabetic or non-diabetic, so all these different, and there are clear differences in the composition of the microbiota. And typically it's more pro-inflammatory sort of taxonomic profiling. The interaction with these, the parasite. This I don't know. This is less clear. I mean, the parasite, in the case of human, when your adaptive immune system is fully functional, you normally control those. But there's clearly, but they could be, for example, I think there could be even triggers of IBD, for example. If you have some susceptibilities that make you maybe more sensitive to some infections, they might switch on an inflammation, and then you don't regulate it properly, and then you get problems. But in terms of taste, I've got no idea. But I suspect there might be an answer towards the gut-brain axis that might be involved. I wouldn't be surprised with that. Yeah.
Okay, thank you very much for a very nice and clear presentation. I have just a question. Is there any relationship between this microsporidia and any neurological disorder?
Urogenital?
Neuroinflammatory or
yeah. I don't know. I know that I mentioned the rabbit early on. I know that rabbits get really bad neurological symptoms. On the they get bad symptoms in terms of how the brain function doesn't work anymore properly. So there are definitely issues. I suspect that in the extreme case of HIV/AIDS, when you can have systemic infection, and that can end up in the brain. So there's clearly cases like that. You can find literature on that, but that would be extreme cases of being immunodamaged in extreme way. Okay. As far as I know, there's no other context.
Okay.
But to be checked. I haven't checked that.
Thank you so much.
Ci vediamo qua. Ok?
Oh, fantastic. Ok, so parte 2. Should I start? Yeah.
Qualcuno si lamentava, io non so come si alza. Start this way. See. It's so complicated. Okay.
You want me to get out?
Yes.
I
No,
do-
to put it a little bit.
further up. I don't know. Maybe I'll take it. You. Can you hear me, guys? Is that working? Can you hear me, guys? Yeah? I keep talking? Okay. Okay, so now is the real reason why I'm here, right? Is to talk about trichomonads, which is, oh, I like that. There's a little magic corner.
To avoid people like me going
So this is the group of parasites that brought me to Alghero. No, it's okay, I use that. Don't worry, yeah, it's too complicated. And my initial interest about trichomonas was from an evolutionary perspective, like the Microsporia. And then as I started to study them from an evolutionary perspective, slowly, gradually became more interested in their pathobiology, which brought me to interact with you guys. And so I'm going to talk about some of the data we've generated in collaboration with you, just a little bit, because you know most of it anyway. So, but this, maybe the audience, some of audience don't know. So we're now going to shift from intracellular parasite, Microsporia, to extracellular parasite, okay? So they don't go inside cell, although sometimes they do kill cells, okay? And to feed on them or whatever is coming out of them. But again, we're going to look at, in particular, the importance of, and I think it's probably one of the most fascinating parasites. I'm sure Gigi and Paola and Tina will agree, because we're going to see different layers of involvement between microbial eukaryotes, bacteria, some of them intracellular, some viruses, and then a whole range of complex member of the microbiota. And these different members that are all part of the same environment can have interesting impact on the host. Okay? So we're going to look at the interaction between bacteria and trichomonas, and then how that may be modulated by viruses. And we're going to see how those different dynamic interaction in the context not only of human infection, which was what brought me initially with T. vaginalis, a sexually transmitted parasite, but then also later on, I will try to convince you that it's very important and very interesting and very useful for research purposes to also look at organisms that actually closely related, perfect, I think that's better, yeah, that are closely related to these ones, but in fact completely different hosts in different sites, and that would be the pigeons, okay? All right, so let's keep moving. So extracellular parasites, you find them in human, find in animals, and as I said, if I do a good job today, my aim is to convince you tomorrow you will be working on pigeons. All of you. Okay. So trichomonas, you can find them in lots of different sites. I already mentioned T. vaginalis in the urinary tract. There's also the oral one, Trichomonas tenax. And we're going to see in particular how interesting it is, because T. vaginalis is basically a human-specific organism, okay? But that's not the case for tenax. Tenax, you will find it in lots of different hosts, including in humans. So we're going to talk about that in a bit more detail. You can also find sometimes in the lungs. HIV/AIDS patients with messed up immune system have more Trichomonas more often lungs infection with Trichomonas. You can also find relatives of them in the digestive system. We're not going to talk about them. We're going to focus on the genus Trichomonas. Okay, so Trichomonas vaginalis, STI, so dependent on intimate interaction. It's a really interesting organism. It can change morphology very quickly. There's not many organisms that can do that as effectively. Here are nice, beautiful pictures from Marlene Benchimol, a colleague in Brazil. These are from biopsies from patients. These are vaginal epithelial cells to which the parasite is attached to. And then if you grow it on substrate, it will tend to form pancakes, amoeba forms. And if you grow it in the tube, a lot of people here do that on a regular basis. We do that as well in our lab. You see this flagellated form. So they can, a trophozoite, okay? It can eat other things. He's eating yeast, for example. And then he has another form that is sort of called pseudocysts. They form little spheres. And potentially these are relevant for infection. They're a bit more resistant to environmental conditions. Their biology is poorly known in general, okay? So I'm going to talk mainly about organisms that are in the trophozoite stage that we can grow in vitro, and we study them in the context of interaction with other organisms quite a bit. Is that working? Oh, yes, it is working. Oops, sorry, guys. So now one of the things that started me to be very interested in animal infecting Trichomonas is this kind of phylogenies. The red lines here are isolates from humans, okay? And that includes, of course, Trichomonas vaginalis, all right? The blue one are isolated from birds, okay? Now, that was very intriguing to me. I was looking at these trees and said, infecting humans, pigeons, and mostly pigeons, by the way, okay? So we can start to have a little introduction to pigeons. And then I was checking the literature, and then nobody was really discussing this. I said, what is going on here? We've got human infecting parasites. We've got closely related phylogenetically organisms that come from pigeon, and nobody is discussing that whatsoever. So what's going on? So I got really interested in that, and that includes Trichomonas tenax, very closely related to Trichomonas gallinae. That's the classical one that if you work with pigeons, if you work with birds and so on, many people will know that, okay? So very intriguing. And that led, and they are, for example, for the Trichomonas tenax, this is a very interesting one. We're going to see that you find it in cats, in dogs, but you also find it in pigeons, or very similar in terms of sequences. And you can also find it in a range of other birds in the cloaca. This is oropharyngeal samples, and this is the cloaca from different birds, okay? It's all over the place. So what's going on here? So that led us to write this review. I had it all planned up. Then I had too much work. I didn't know what to do. And then I bumped into Jane Carlton and said, oh, so we had a discussion with Jane Carlton is the one that sequenced the first genome, and then subsequently more genome and everything. They did a fantastic job for the community on Trichomonas. Without those genomes, we'll be having less fun. Let's put it that way in terms of the science. So, and she said, oh, I've got this really good PhD student. She writes beautifully. So we had the plan. Everything was set up. She's also a very good scientist. So everything was perfect. So we wrote this together, and that got published here. And we basically proposed sort of a general model of nothing, no, it's not nothing fundamentally new or anything, but it was never properly discussed. And it was about where does the Trichomonas infecting human come from, okay? And in particular, it was intriguing that so many lineages from isolates from human, tenax or vaginalis, were so closely related to isolate from birds. So clearly birds, you know, caught my attention. And then if you look at other species, tenax, if you look at literature, you find them in cats and dogs. So we're going to come back to that a bit later. I'm not going to talk about the others, so we can just move forward. Pigeons. Okay. Now, pigeons, many people think this is boring. Some call them rats with wings, dirty. They mess up everything. Why would you look into pigeons, okay? Now, I happen to have at home the volume 4 of Handbook of the Birds of the World. It's an amazing book. It has many volumes, but I only bought the first 4. Then we had 2 children, and I was not allowed to buy any other volumes. Okay, my wife was very strict about that. So, Alina, as you know. So, but I was lucky to have volume 4 because it covers the pigeon. So I said, okay, let's have a look what we can find about pigeons. You don't need to learn that. The point is, there's lots of different kind of pigeons, okay? The one that we are more familiar with are the Columbinae, but you've got some other guys a bit more exotic. Some of them are gorgeous, beautiful pigeon, beautiful birds. This is from New Zealand. The locals call it the keruru, apparently. It's very important in the Maori tradition, also for food, when they have important meals. Then you have this is from Comoro Islands, really beautiful pigeon. There's lots of other ones, very colorful ones. Just one more, and I will stop here. There are more. Okay. So if you like birds, have a look. Yeah. Unfortunately, you have to travel far away to see them because it's not around here. But anyway, just to point out this nice. Now, why do I mention that? Why bother about pigeon diversity in addition to the fact that they're just beautiful and colorful? Now, an Australian group decided to look at some of these cool-looking pigeons. Okay, so it was realized that the few people that actually bothered looking to this discovered that if you take isolates from different pigeons, you sometimes bump into something new that is not Trichomonas gallinae. In terms of Trichomonas gallinae, if you walk outside this campus, there will be the rock pigeon, there will be the wood pigeon, probably. In the UK, it's the most common one, is the wood pigeon, the big one with the white. 40 to 60% of them have Trichomonas gallinae. It's everywhere around you, okay? Now, in addition to that, there were a few papers that suggested some more Trichomonas here and there. So they decided to systematically ask the question, okay, we're going to go, and there's a huge diversity of this pigeon, as you can see here, where you have the different species and how they map. Some in New Papua Guinea, the New Zealand, the Keruru one I showed you earlier, very nice. And then a range of in Australia, and they also had a sample pigeon from Africa, okay? So, do we find Trichomonas? Yes. And not only that, but they basically doubled the diversity of known Trichomonas by doing that. So the prediction was right. They thought, we probably find new things. So they find lots of them, and the idea is that the data they got from phylogeny, biogeography, and so on, is that probably the Trichomonas appeared in pigeon and diversified in pigeons. Now, look at this phylogeny. This phylogeny here shows this is ITS sequencing, okay? And each of those lineage that doesn't have a label, no color, is from a pigeon, okay? Pigeon, 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 T. vaginalis, human. Then Brixie, that was found by the Czech guys in cats and dogs. Pigeon, 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 pigeon all over. Tenax, oral cavity of dogs, cats, and humans. Pigeon, pigeon, and then all rest pigeon, okay? The long branch here is a nice one from Keruru. It's not too surprising. If you look at geography, New Zealand was separated from the rest for a couple of million years. So there was a pigeon that differentiated from all the other pigeons, has a very distinct morphology and so on. And the Trichomonas evolved with it, and is very different from the others, okay? So nice evidence for coevolution. But what is really fascinating on this tree is that you've got all these different pigeon things, and from time to time, bing, you have something appearing in a mammal or in a human. The simple explanation for that is that you have jumps from pigeons to mammals, including our STI, T. vaginalis. Very intriguing, okay? So when this was all clear, I said, okay, now we definitely need to work on those pigeon-infecting birds, pigeon parasites. Now, the other thing in terms of the biologist here, if you're interested in environmental issues, or if you are an ornithologist to some extent, Trichomonas gallinae that you found in pigeon, here's an example of the wood pigeon in the UK. You probably have the same here, no? We definitely have in Switzerland, so I suspect it's probably in Sardinia as well. Now, there are some strain of T. gallinae that are very virulent for other birds. And if they get transferred to greenfinch, chaffinch, here's one example of both. This happened in about 2000, early 2000. It was associated with collapse, I mean, really bad collapse of their population. So really dramatic. This is associated with a strain called A1, which is more virulent than the other one. There's a whole range of strains that people identify. So really relevant. And they even decided to, now you got, in the UK, they're very fond of their birds. I think here they're associated with birds a bit different. They like to make soup with some of them, at least. Anyway, so if you're interested in keeping your birds healthy, you should not feed them, because that's where the transmission goes on. If you put food in the garden for birds, the pigeon will turn up and will contaminate they've shown that, it's all demonstrated, okay? So if you're interested in birds, do not feed them in the summer for sure, and clean the place. So that made me very interested in using comparative approach to better understand the biology, not only by studying T. vaginalis on itself, which is interesting, of course, but also looking at T. gallinae and also T. tenax. What do these things have in common? Can we find features that are fundamental to their biology and that may explain this? You know, how come, oops, sorry, I'm not very good at this button thingy. How come that we have all this multiple transfer from, it looks like the same source? So there must be something interesting there going on. So I was really interested and fascinated by the idea of, let's systematically compare T. gallinae, because there was a genome, draft genome out there, tenax, there was also a draft genome, and of course, long before that, there was a draft genome of vaginalis, okay? So being interested in the evolution of parasites, their genome and the genes, that was an obvious thing to do. We couldn't resist. Okay, so what did we do? By the way, in terms of diagnostic, tenax can be found in urinary tract sometimes. There's a paper in Israel, homosexual relationship, and they found there was misdiagnosed as vaginalis, and it happened to be tenax. So another site. Anyway, so little parenthesis. Now, what is really interesting about Trichomonas vaginalis, this is the best studied one, okay? Including by you guys, obviously. And here we have some nice set of collection of papers that illustrate the relationship between infection by Trichomonas vaginalis and change in the microbiota. So when you have Trichomonas vaginalis, you don't see exactly the same profile of bacteria present in the urinary tract of women, okay? Nothing is known about male. No data. We should look into this. Nobody has. Anyway, so the point is that there's lots of variation, and in particular, this is where Gigi and Paola, and also historically, I think the Mycoplasma is you guys, right? 100% you. You were the first one. So, and it's a Lancet letter, right? So this is very interesting. So one of the things that is absolutely amazing with Trichomonas vaginalis is that not only it interacts with members of the microbiota that are extra outside itself, but it has also some symbionts, under symbionts, okay? So another layer of complexity. And I will briefly mention some of that, how it's relevant for the biology of Trichomonas. Of course, that's data from here. And we're lucky enough to be able to collaborate with you, to have a lot of fun research and very exciting data. So I'm going to mention briefly. The point I want to make, sorry, I forgot that, is that here there are clear numbers of examples, and T. vaginalis is one of them, where you have a strong relationship between the presence of the parasite, or microbial eukaryote, when you have dysbiosis due, for example, loss of the mutualist. So in the urinary tract, what's the most important, women, what's the most important mutualist bacteria? Anyone knows? Not you, obviously. Anyone knows in the audience? I didn't hear anything. Which bacteria is very important for women urogenital health, including reproduction? See? Come? Lactobacilli, exactamente. Okay? So, when you have Trichomonas vaginalis, you typically lose lactobacillus. They are gone. Okay? Trichomonas happens to love eating them. Okay, we're going to talk about that a bit later. Okay? So that's an example of how you can modify the composition of the bacteria, in that case, by probably specifically targeting them. And I will show you some new molecules that we found that may contribute to that as well. Okay? Shared by all the Trichomonas. Not only T. vaginalis, but Gallinae and Tenax. Okay? Okay, so This is now the model people understand. It's the work from Gigi, Paula, others as well. We have contributed a little bit to that, but most of that work indicates that the inflammation, the thing that causes disease, that can cause disease and problems, is a complex combination of the parasite alone, or in combination with endosymbionts, Mycoplasma, 2 species, different, and potentially also viruses. And this is associated with a modified microbiota, which is typically called bacteria vaginosis, or the overlap of species between what you find in T. vaginalis and bacteria vaginosis. You don't need Trichomonas to have bacteria vaginosis, okay? But the combination of bacteria overlaps, okay? So you have clear changes, modification of your microbiota, and this is associated with increased inflammation, okay? And increased damage to the host tissue. And that's one of the reasons there's a strong association between T. vaginalis infection and other infections, such as HIV, for example, okay? There are really clear epidemiological data indicating epidemiologic synergism between T. vaginalis, HIV, some data about HPV as well, and so on, okay? So a really interesting example where we have complex interplay between microbiota composition and the symbionts, which make Trichomonas unique and so fascinating in many ways, okay? So what about the bird infecting one? So we were going to do this experiment described in this paper. Nick Bailey, a PhD in my lab. So we said, okay, we're going to get some pigeon sample. We're going to compare infected and uninfected. And then one morning we woke up and this group in China published this paper where in China, they produce billions of pigeons for food, okay? So it's a traditional part of the diet, including medicinal impact, according to them. It's a bit like us, at least in the UK or Switzerland, if you have flu, you're told, eat a soup of chicken, right? Same idea, all right? So a lot of pigeon is produced for meat, because they like it. It's actually supposed to be quite healthy. I never eat pigeon. I'm not interested in the idea because, flying rats and all of that. But anyway, I was told it's very good by the French postdoc, Johan. I asked him because French people are the #1 producers in Europe of pigeons. Now, what did the data show? They show that, like in human, you lose lactobacillus. So this targeting, or at least this relationship between presence of a Trichomonas and absence of lactobacillus is shared between T. vaginalis and Trichomonas gallinae, which I found very interesting. So that's one interesting point. And what about Trichomonas tenax, the one that is the oral cavity? So this one you can find in dogs, cats, is also associated, like in human, with periodontitis. Okay? Periodontitis, a little reminder, is when you have an excessive inflammation of the soft tissue initially, and then it gets bad, worse, typically, and you start to damage the bone. And the outcome is that you're going to lose your bones if you don't go to the dentist. Okay? And that is an inflammation that does not need tenax. It's bacteria-driven. But, as we just discussed, in pigeon, in human urological tract, we know that there's an association between the parasite and bacteria that are pro-inflammatory and so on. So the question is, to what extent is maybe Trichomonas associated with that? It is associated. How? What's the link between the two things? People haven't figured that out. So we were interested in trying to figure out what's the molecular basis that is shared between tenax, gallinae, vaginalis, and how they interact with the bacteria, and how that may explain inflammation in T. vaginalis, inflammation in pigeons, and inflammation in oral cavity of dogs and humans and cats. So that's where comparative genomics comes in. Okay, and remember I showed you earlier some work we did initially to look at lateral gene transfer, and we had some really interesting genes, and we're going to come up to some of them now. So one of the cool things by having access to all these different genomes, we can ask the question, okay, we have an interesting gene that we found in T. vaginalis. Is that shared with the others? Okay? So that's one of the things we got very excited about. And so here we're going to look at a little bit about Trichomonas mycoplasma interaction from a comparative genomic and transcriptomics perspective a little bit, and we're going to have some data that we bumped into by chance that shows that one of the mycoplasma that you find in human associated with Trichomonas vaginalis is also found in pigeon. So what you guys found in human seems to exist in pigeon as well. Okay, so very interesting, I think. And then we're going to say a bit about Trichomonas interaction with fungi and bacteria and look at some of the molecular detail associated with that. Okay, so what we've done, well, historically, the first thing was your work with mycoplasma, and we were interested when you were showing us this fantastic result of impact, dramatic impact of how the parasite behave in presence of mycoplasma. We decided to collaborate, and it was very exciting, and we got amazing data. So we are just very briefly mentioned that. But then we wanted to extend that to other systems, and also in particular look at extracellular organisms. How does extracellular organism modulate Trichomonas, and in different species, ideally? Okay. So Adam, I will show you some data about Trichomonas vaginalis interaction with E. coli. Lenchina did comparative genomics and also studied Candida. And Isra is currently working, we collaborate with Honza to look at Tenax and other species that are isolated from neural cavity. Echibococcalis, I don't think I pronounced it properly, but from horses, yeah? And the other one is called Brixie from cats and dogs. So we're looking at these things from this different perspective. It's quite dramatic. So in the same way, the work from Gigi's group, Valentina, Paola, have shown that there are many genes that get differentially modulated by the presence of different mycoplasma. You get the same thing when you do incubation with E. coli or with Candida. Okay? I'm not going to go into detail much. I'll just highlight, I'm going to skip that because otherwise we'll spend the rest of the night here. So this is the work we did. So there was a really nice collaboration. It was very exciting to be involved with that. Valentina's work, Nick Bailey was a fantastic PhD student in the lab. So they worked together to generate this very interesting data set that aimed at understanding how does Mycoplasma hominis or Mycoplasma giardardi, they renamed those species now. I'm not going to go through those names. They have different names now. It's a bit boring, but anyway. And how do they modulate? Do they modulate anything relevant? Okay. Now, Gigi was always very interested in hemolysis, so had nice data showing that different strain can be more or less aggressive. Okay. And Mycoplasma do stimulate the key genes that are thought to be behind the capability for Trichomonas to do that. And those Mycoplasma were indeed stimulating, I guess these are your data, Valentina, yeah? What about cell adhesion? So that's one of the things I was always very interested as well. They have this enormous collection of very strange genes that code for surface protein that look like bacterial genes. There's loads of them. And we looked at a whole collection of them. And again, as you can see the different color pattern here, many of those genes are significantly modulated by the presence of one or the other or both Mycoplasma. So Valentina did a wonderful essay. Is that right? Yeah. Just to make sure I don't... And so the prediction was, based on the comparative transcriptomics we have, probably there will be a difference. And not only that, the difference was massive. Okay? So the way the parasite interact with the host cell seems to be significantly modulated by the presence of the endosymbiont, which is really interesting data set. So really fascinating organism that has not only complex interaction with the host, epithelial cells, immunocytes, but also interaction with the microbiota and its endosymbiont, and they influence each other. There's lots of other things happening that we're not going to cover here because otherwise, as I said, we're going to, in terms of metabolism, for example, that are modulated as well. So that's the model now where we know that these guys here, Mycoplasma hominis, Mycoplasma giardardi, are really, the important players. And ideally, diagnostic of T. vaginalis should be not just T. vaginalis. No. Is it T. vaginalis with TVV or with hominis or giardardi or both? And you have shown that the presence of both is quite common, including in Italy, in the samples that you got. So it's a really clinically relevant thing, potentially quite relevant, and therefore one of the first things that should be done, I think, is change the diagnostic to say, oh, it's TVAG this, TVAG that, not just TVAG on its own. Okay, so now, as I said, I've got obsessed by pigeons, okay, and also other animals, cats and dogs. And because that's so closely related, remember the phylogeny I showed you earlier, I'm interested in trying to compare what is going on here, T. vaginalis, tenax in the oral cavity, urinary tract, and how does that compare with what's going on in pigeon infecting or other animal infecting Trichomonas, okay? So what do they have in common? We know, I already showed you that we know they have some commonality. For example, lactobacillus tend to disappear in both T. vaginalis and gallinae. So what about at the molecular level, can we find genes that are shared that may tell us something about that? And why, for example, they jumped so easily from pigeons several times to mammals? Yeah? Why? And how? So the first thing was an accident. So that was really exciting. So Nick, that was part of the preparation we had to do experiments to do the microbiota. And then we also decided to, let's try to do transcriptomics. Can we learn something about the transcriptome of a parasite in vivo rather than in vitro? Okay? So we took swabs. We went to visit a guy that is called pigeon fancier. These are the people who do racing pigeon. I'm sure they're here as well, no? So they're obsessed with pigeon, and they will travel with trucks, and they will let the pigeon go, and they do racing, and they win prizes and things like this. I have no idea why would you do that, but some people like doing this. So, but for us, it was very nice to talk to them because they were very keen to know which pigeon have parasites, and we were interested to find the parasite in the pigeon, so we have potentially data from in vivo. Okay, so we took a swab. When I held the pigeon, and Nick took the swab. And we did that for several pigeons. We had a collection of them. And in one of them, we did a metatranscriptome. Can we find reads for the Trichomonas? And can we learn something of that? The experiment was a disaster. The problem was the kit that you normally need to destroy the ribosome RNA didn't work. So we paid a lot of money. I was really annoyed with that. We paid quite a bit of money for that. But there was something very interesting that turned up. So we identified a full-length, large ribosome RNA gene from a mycoplasma, and it was very, 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 very similar to Mycoplasma gerardi, the one found in humans, okay? My first reaction was, Nick, what have you done? You contaminated the bloody samples. Yeah. But no, it was not, because the sample that he got from you was analyzed later. So we had those in the fridge, in the freezer. They were there, but he didn't touch them. And he analyzed that before. So there was no possible contamination. And of course, there were some key distinct differences that clearly demonstrate it's not the same thing. So the phylogeny tells us that there's a mycoplasma in the pigeon that is associated with Trichomonas gallinae. And what was interesting is that, we know that thing exists and nobody else knows. So we went back to the Chinese data. Remember the paper I showed you earlier? So Nick reanalyzed that data set. They did mention mycoplasma, but they didn't look into details. And they had no idea about this one. So we checked, and bingo. There's a bacteria, it's that one, strongly associated with infection. So what you have here is a data set where you have pigeon with Trichomonas gallinae and pigeon without. And you can compare. And you can see, for example, that you lose the lactobacillus. So the red bar is that you're losing the signal, okay? Whereas the green one are enriched. Prevotella, does that ring a bell? It's the same in the urinary tract of women, okay? So when you have Trichomonas vaginalis, you often have Prevotella as well. You see the same thing in pigeon. And you have this mycoplasma that is also very strongly associated with infection of T. vaginalis in humans. So we've got exactly the same profile, really fascinating. So I'm not going to go into further detail, but that's just to say that basically we've got a similar picture here, what we see in humans, in pigeons. The pigeons are a really cool model system now, I think. We sequenced the genome, so we collaborated with the Chinese. We contacted them because they had tons of pigeons, tons of material. We only could get 1 or 2 pigeon samples, And there was also COVID. It was a mess afterwards, so we couldn't get, the guy had health issues. So we contacted them, and they were very happy to collaborate. It was brilliant. So what they did, we told them, look, can you get a swab and take a swab from a pigeon that has high numbers of reads for Gallinae and that is positive for this bacteria? So they did that, and then they did metagenomics. So you just sequence everything. Yeah? And because the Mycoplasma genome are small, bingo, we had a complete genome. There was a tiny gap. We did the PCR, finished it, all done. Complete genome from the Mycoplasma coming from pigeons, and it's essentially looking like the human genome. There's some recombination, reverse, and so on. But it's a very closely related species. It's actively dividing because when you count the numbers of reads away from the origin of replication, you can see the numbers going down. So that's consistent with a genome actively replicating. So it's a Mycoplasma associated with T. gallinae actively dividing in a pigeon. Okay, so it's live. It's not just a contamination. And what else? We could also show that it seems to be kind of co-evolving, because we found some other. Nick did a fantastic job. He went to the literature very carefully, and he found some other weird Corruga. This is a genus of a trichomonad that you found in termites. Okay, and they isolated the Mycoplasma from it, and it's part of the same group where you have these guys. So it could be, and I said could, keyword, I'm not saying it is, evidence for long-term coevolution between trichomonads and Mycoplasma. Okay, so very intriguing. Now, this is a data set I just saw. The other thing that is interesting with those genomes, they encode homologs of one of the protein families I showed you earlier. I didn't mention them, BSPA. These are surface protein that were shown in bacteria in the oral cavity to be very important for bacteria-host interaction and bacteria-bacteria, including Treponema and Tanarella. Now, look at this. These are all full-length Mycoplasma genomes, okay? We did the phylogenomics. And the one in green are the one that have BSPA homologs, okay? Now, who has that? Mycoplasma gherardi, Mycoplasma trichamica, the new that we found it. And I challenged Nick to come up with a name, and he said trichamica. They like trichomonas. And then we have Mycoplasma hominis. How interesting. Now, if you do blast against all in all, and you look at a network, the network tells you if two sequences are similar, the dots would be side by side. Okay? If the sequences are very different, they would be distant from each other. Okay? And what you do, you connect the one that are more closely related using certain cutoff. So you can see that all the BSPA from Trichomonas trichamica, Trichomonas gherardi, come together. Yeah? But they also come together with the BSPA from Mycoplasma hominis. But they're not closely related. So the question is, how do you explain that? Any idea by the audience? How could you explain that these organisms that are not very close, they're not that far, but they're not side by side, right? They have quite distant. And you have only BSPA here and BSPA there and some others here, but these ones are quite different. You see there's cluster away. But the Mycoplasma hominis and gherardi and so on, how could you explain this clustering of genes? Any suggestion by the audience? Or you're already hungry and need to eat, or need a coffee or something? Suggestions? Gigi? Valentina?
Maybe with the transfer.
Gene transfer.
Yeah.
Yeah. And maybe it happened in Trichomonas because they're all together. So I'm not saying this is what happened, but it looks like it. It smells. It looks like and it smells like it. So very intriguing. So maybe there's some interesting coevolution that happened between these guys, relatively recent. Okay? So comparative genomics is amazing. It's very interesting. Anyway, so just to summarize this bit, I think I mentioned all of that. And for the sake of time, since we're all getting hungry and tired, including myself, the point I want to make, so support the model of coevolution between Mycoplasma and Trichomonas and co-zoonotic infection. That's the thing I find interesting. The simple explanation to have Trichomonas Giardardi and Mycoplasma giardardi and T. vaginalis associated, and Trichomonas trichamica with Guyanae, and so closely related, is that they have co-evolved. Yeah. And it was a co-transfer from an ancestor that had both. It's a simple explanation. You can come up with more complicated ones, but so the symbiosis could have contributed to facilitate zoonotic transfer from birds to mammals on humans including. Okay? And we know that the mycoplasma modulate how effective they bind to tissue. Yeah. So maybe they needed that. I mean, it made it more effective to jump host. Okay? Now we have some evidence that not only host is stimulated, but also interaction with bacteria gets stimulated. We'll come out in a moment. Okay, so that's the little story about Trichomonas guyanae. Pigeon for us are becoming more and more interesting. I hope I convinced you as well. And now the other thing we were kind of interesting is looking at, okay, we know that Trichomonas interact with the mycoplasma. We know they are associated with change in the microbiota. So how could we explain change? Does that suggest direct physical interaction between Trichomonas and the bacteria? And if so, what's the molecular basis for it? Okay. So for that, with Lenshina, a PhD student in the lab, she's now in the US, she worked on annotating carefully the draft genome for Tenax, okay? And then we did comparison with Vaginalis. One of the big questions for Tenax, is it potentially a pathogen or not, okay? We asked that question specifically, well, from different angles, but including in particular, is there anything shared between Vaginalis and Tenax in relationship to how it might interact with bacteria, okay? So we found lots of things. First, we found lots of BSPA proteins, which might explain interacting with the host, but also interacting with bacteria. We found the saplip protein that you guys have a lot of interest in, right? So they could do exactly the same thing as T. vaginalis does. We found lots of the virulent factors, different peptidases and so on that are known to be involved in damaging host tissue. And then we found an amazing collection of what is called K-zymes. K-zymes are carbohydrate active enzymes. There's a very nice database done by people in Marseille, the KZ. And we collaborated with one of the KZ guy, the big boss from that place. He's now retired, but still works. And he annotated that with us, helping us to get this done. And here's just a collection of some example of glycosyl hydrolases. These are enzymes that cut complex sugars into smaller ones, okay? Really important in many, many contexts, including in your gut. So most of the things we eat, if you eat an apple, a salad, and so on, you've got no enzyme to digest that whatsoever. Nothing. It's all done by the gut microbiota. So the diversity of K-zymes in the gut microbiota or microbiota in general is usually quite spectacular. And we humans have a ridiculous small amount of K-zyme. All right? So for us, it was very interesting to look at this list. And talking to people who work on those was also quite interesting. And for example, we picked up, there was a couple of enzymes that are likely to target mucus. Bingo. Mucus, targeting, destroying the mucus is quite interesting for a lot of organisms. And also, even more interesting in relationship to interacting with other microbes, enzymes that can degrade the cell wall of fungi or bacteria. Ha! Now it's getting really interesting because now we're going to start to have some specific features that tell us potentially how Trichomonas vaginalis, Guyanae, Tenax, etc., can target the cell wall of different organisms. That is relevant not only potentially to explain how they may modulate specifically microorganisms, but also to generate inflammation. The moment you start to make fragment of cell wall, you generate very strong pro-inflammatory molecules. So it's relevant in those two contexts, okay? So here's I just put a list of things. These are all lateral gene transfer from bacteria, okay? I'm not going to go into details. I will mention that one. These are K-zymes, and I also list the peptidase that a colleague in New Zealand decided to work on that we identified as lateral gene transfer. So we're going to talk about this because they are likely to be functional partners of this one and this one to deconstruct completely the cell wall of bacteria, whether gram-positive or gram-negative. And we're also going to talk about GH30, because that one seems to be involved in degrading fungal cell wall. And finally, we're going to talk about bacteriocins. What are bacteriocins? Any idea? Have you heard of that word before? Bacteriocins. No? Yes? No? These are antimicrobial peptides produced by bacteria to kill other bacteria. Okay? We found that in Trichomonas. Okay? Really interesting. So, Trichomonas express a bacterial bactericins, a homolog, and we're going to see what's the potential relevance of that. So, the model we're going to start to put together is that we have Trichomonas expressing different enzymes, antimicrobial peptide as well, we're going to come to that later, and these enzymes could target the cell wall of bacteria, peptidoglycans, or bits of the cell wall of fungi. I'm going to show you some experimental evidence to support that idea. So let's start with the fungi bit. So Lencina co-incubated Tenax with Candida. They're often together in the oral cavity. And we were interested because benchimol had data showing that Trichomonas vaginalis can eat fungi. So Lencina did quite a bit of work on that. And they can actually interact quite dynamically with both hyphae and the yeast forms. So that was quite interesting. And Lencina did the transcriptomics on that. But what we were particularly interested in is to test the idea if Trichomonas tenax could target the cell wall using the GH30. If we can do that, it would imply that it can expose very strong pro-inflammatory molecules, which would be directly relevant to understand vaginosis and periodontitis. So, GH30. So, it was very interesting. I walked down the corridor, a colleague, Liz Lowe, characterized this enzyme in Bacteroides. It's that one. Okay? So I went to see her. I said, look, we have this GH30 from T. vaginalis, T. tenax, T. gynae. And how does the sequence look like to you? So we had a look together, and she said, oh, boom, boom, boom. It has all the key residues that makes that GH30 from Bacteroides a very specific enzyme that targets what? That targets the, wait a sec, yeah. So these are the structure from the Bacteroides gene, a protein, sorry. And then Trichomonas has all those key residues. So all the residues listed here that are essential for the activity in the Bacteroides to target the cell wall of fungi are completely conserved. So even before doing the experiment, she said, this is the same thing. There is no way, from a structural perspective, it's going to do exactly the same thing. So it was very exciting. So we tested that. And bingo. So if you use, it's basically targeting beta-1,6 glucan. Okay? So you can use the Bacteroides enzyme. You can use different pH. It works for a broad range. If the pH is too extreme, you start to be having problems. And then you can see the different fragments because the enzyme is not able to fully degrade the complex sugar. Okay? So what you get is basically from beta-1,6 glucan, you get glucose. And what do you do with glucose? Glycolysis. You eat your pizza, your pasta, no? Lo mismo. Excellent. But very interesting and relevant for inflammatory disease. Once you target beta-1,6 glucan, there's not a lot of them. But the problem, when you do that, you remove the mannan part of the cell wall of fungi. And once you have done that, you're in trouble. Well, the host is in trouble. Because the beta-1,3 glucan are mega super duper pro-inflammatory molecules. Okay? So not only can Trichomonas target the cell wall, possibly have a nice lunch, nice pasta dish type thing or pizza, but it will, as a side effect, it will actually generate pro-inflammatory responses as well. So potentially quite relevant in lots of different. They all have it. Tenax, gynae, they all have it. Okay? So that is this story here. So we have some clear case of an enzyme targeting the cell wall. What about bacteria? So that started with Augusto. He's from Brazil, but works in New Zealand. And he contacted me and was interested in kind of, for me to do the bioinformatics, because we had actually the phylogenies that looked at the origin. These are bacterial genes. that encode peptidase that are thought to be very specific, at least the bacterial one, to degrade peptidoglycan, the peptide bit of peptidoglycan, all right? So 2 papers came out from this. So we did the phylogenetics, I played with that. The key thing to show here is that all trichomonas have it, vaginalis, gallinae, tenax, and you had 2 different acquisitions from different group of bacteria. So you have 2 group of families, and each of those families have members in the 3 species. And they are all monophyletic, so that means it was acquired by a common ancestor. So the phylogeny are interesting in that respect. The alignment, what the alignment shows is that they all had the key functional residues. So from a bioinformatic perspective, they're all functional. Okay? And Augusto went to test that. Then he couldn't functionally characterize them. So he asked me, do you know anybody who? I said, yes. There's this guy around the corner in Newcastle. If a Martian comes to planet Earth and wants to know how bacteria deal with a cell wall, you speak to that man. Okay? And so Valdemar, I contacted him, went to see to his office again, like Liz early on, and he said, oh yes, tell Augusto to send me the stuff and we characterize it. Because that's what they do day in, day out. So, and they could demonstrate that this peptidase is indeed exactly that. It targets the peptido bit of the peptidoglycan. What he could also show is that if you overexpress, this is the work from Augusto's lab, if you express the wild type, or one of the wild type enzyme, you destroy E. coli population, okay? If you overexpress a mutant where the key residue, catalytic residue is mutated, you don't see that phenomenon, okay? But what is interesting, if you put the enzyme purified in a tube, zilch, nothing happens. E. coli doesn't care. So it needs the peptidase plus other things. What are these other things? So that's the things we looked at. This is to show that it works on lactobacillus. So Trichomonas has peptidases. You overexpress them from both sides. Do you remember the 2 clans, clan A and B? So both members are very effective at targeting the cell wall of lactobacillus to such an extent that you start to impact the population. Okay? So if you have nothing in the vector, you have this, and if you overexpress, you can see the colonies, CFUs, going down. Okay? Now, what about this other GH19, for example? GH19 are interesting. These are glycosyl hydrolase that can do 2 things. Either chitinase or lysozyme. So that's quite an interesting thing in the context of this discussion. So Adam decided to test that. So I'm going to show some data about this. So I'll show you that this works. Okay. Augusto showed that this works. And now we're going to look at asking the question, can we have other enzymes that target the sugar bit, the glycan bit of the glycopeptide? Okay. So, first, sorry, let me talk about Tenax. Yes. So recently we also started to work with Tenax more specifically. And here's the guy I mentioned to you guys, the Canadian dentist, Marc Bonner. You can have the link and everything. And what he does, he trains dentists to recognize Trichomonas tenax and Entamoeba gingivalis using a microscope. It's not very subtle, but it works. Now, Tenax, Takezi, Jan Takezi, shown that you can find it. Here's a cat with nasty periodontitis, and they have lots of isolate from both cats and dogs, and also horses, by the way. So we are collaborating, and Isra, PhD in the lab, that's Moody we had early on with the Microsporidium. They work nicely together on complete different project, but you need to have team beside you. And here's a little movie that shows. This is a movie actually from. Can I make it work? No, I cannot make it work. How does that work normally? No. Okay. It's a little movie that comes from the Canadian guy. And it actually looks like what I look like when I go to Takezi's lab to look on the microscope. The Tenax swims around, and the bacteria are swimming around as well. And the Trichomonas just goes around and has a try to taste it. Anyway, so what we have done is that we took advantage of these xenic cultures where you have this amazing high density of Trichomonas tenax in combination with all the quilombo, the bacteria. Okay, complex mix of bacteria, and we did metatranscriptomics. You asked me to use the word, so I thought that was the good time.
Yeah.
So, what was the translation? What did you say? Un quilombo.
No.
Anybody can ask later on what it means. It's from Argentina. So, what we have is this very complex culture that has Trichomonas tenax and a complex mix of fungi, bacteria, and God knows what else. So we extracted those samples, extracted RNA, and did metatranscriptomics. So we sequenced everything at the RNA level, okay? After removing ribosome RNA. That's not interesting to sequence. So with that, we can do, on one side, we can map where does this sequence come from. Simply take the sequence. It's Illumina reads, 150 base pairs, I think it was. And then we can get an idea of what organisms are present. So we were hoping to get tenax. We're expecting lots of bacteria and maybe a few other things, okay? And then we also, and we had quite a few reads on that, and we did millions of reads. We did that several times. So we have massive coverage, okay? That works well if you have a lot of sequence, so you get a good picture of who is there, okay? And what genes are there. So I'm going to show you Kraken output, and then I will also show you a few initial data from, I do have a laser, sorry, forgot. Initial data of where you take the reads, only take the reads from tenax, and create clusters, and then you get an idea of what genes they code for, okay? And we're going to look at some of the functional potential relevance of these. Okay, so in terms of the taxonomic mapping, so we had quite a few reads from the eukaryotes, Trichomonas tenax. It worked, okay? So we were not sure, right? So we had quite a few reads, a million reads of Trichomonas tenax. Then we had lots of other bacteria. Is Daniele here? No. Oh, yes. Which bacteria are you interested in? Is it maybe here? So we have
No, it's not here. I forgot about
Porphyromonas, very common in the oral cavity. This is from a cat, huh? From the cat. This you find in humans. It's not necessarily exactly the same species. Huh? I was told, talking to Nick, he told me that they are very similar species, but they're slightly different.
Okay.
But in terms of mapping, because there's nothing known about the cat, it will map on the human one. Okay. So we have very similar sets of species, and in particular, we have Tanarella for Scytha, we have Porphyromonas gingivalis, and I thought we had Treponema, no, but Campylobacter, some other species you find typically in the mouth, okay? So that worked. In addition to that, we had some interesting viruses. Quite a few phages, not surprising. Lots of bacteria, you expect phage, okay? But there was something, most of the reads coming from orthonavirae. These are where the totovirus are. On totovirus, bingo, are what you find in Trichomonas vaginalis, in Giardia, and so on. So that was very exciting. So I'm going to show you first a bit of annotation. I'm not going to go through the detail because it's too late. But basically, we had some high level of confidence for many gene. So these are the number of gene families. Huh? So that means we have many things clustered together that have high level of annotation. Multiple tools agrees. We know we have a good idea of what they're doing. Okay, and then you have some intermediate stage. So what do these things code? A lot of things are for adhesion. So many genes are highly expressed, involve BSPA, PMPAs, and so on. Quite a few on proteolysis, peptidase, and so on. Oxidative stress, so she decided to focus on that. I'm not going to say much about this, simply to say that we have quite a few proteins that we know are important for bacteria-bacteria interaction, and probably Trichomonas bacteria interaction and Trichomonas host interaction, okay? And BSPA in particular is a very strong pro-inflammatory molecule in Tanarella and Treponema, and they use it to interact with each other. So Trichomonas says that, vaginalis, and tenax, and gallinae, okay? So really interesting shared feature. There are also quite a few lectins. So I'm going to briefly go through that so you don't spend too much time on that. Wait a sec. Then thyrodoxine reductase, the peptidase. Sorry, I missed the peptidase. Yes, so there's a lot of peptidases, including, for example, highly expressed. Not surprised to see them highly expressed because there is full of bacteria in the Tenax context, okay? Also, quite an important machinery to oxidative stress response, which you will expect, so that's quite nice. So all of that is consistent with an environment a bit more closely related to what you see in reality when you have actually many bacteria and things around it. In terms of the viruses, we looked at the sequence and we could Isra could reconstruct 2 full viruses. So we collaborate with Honza at the moment with that. We have a TVV1-like virus and TVV3 virus. Now, why are we interested in that? Who cares about the virus that infect the trichomonas? So these are viruses that infect eukaryotes, okay? And why is that of interest to understand trichomonas interaction with the host? Because these viruses, although they don't infect human cells, they are detected by human cells, TLR3, dependent detection, and they trigger very strong inflammatory response. So they're completely irrelevant from the perspective of infecting a human cell, but they are highly relevant in triggering inflammatory responses. So if we have a T. vaginalis or a T. tenax with those viruses, it might actually cause worse inflammatory response than if they're not present. So that's why we're interested in that. I think, next, oh my God. Yes, I forgot that. Next, we're going to, how much time is left? I should stop soon. So I'm going to quickly zoom through this. So with Adam, he did a co-culture, and what he could show that if you have E. coli, you can grow tenax Gallinae for longer. So that's the pigeon infecting one, okay? So you put E. coli, what we think is happening is that the E. coli grow, and then Trichomonas gallinae will eat E. coli later, so can survive. Whereas if there is no E. coli, the population collapses, because all the nutrients are gone. Whereas here, the bigger of the nutrition goes into E. coli, and then Gallinae has access to the E. coli. All right? So that's what we think. Now, what was very interesting that Adam noticed, the Gallinae don't behave the same. They go crazy. Okay? And indeed, they are much more active in terms of speed. So if you do the statistics, they did live imaging. The behavior of Trichomonas gallinae in presence or absence of E. coli is very different. So they sense the bacteria, and then they change the behavior. Okay? Then we looked at the transcriptome for this time point. Do we see a difference when E. coli is present or absent? Like Mycoplasma, absent or present, okay? So we found many genes of interest, but what was also interesting, we were expecting to see what you see with Lactobacillus. If you incubate Lactobacillus vaginalis, it will eat them. One of the reasons you probably lose them. Okay? We didn't see that with E. coli. But what we saw is little pockets. We never set them inside. It's never inside. So we don't see that. But we see them interacting physically. So quite interesting. Then, in terms of the gene, there's lots of very highly modulated gene. The top one is an antimicrobial peptide. One of the top one. Most high. So the genes to be listed here need to be significantly upregulated and expressed at a high level. One of the top one is AMP. The other one is GH19. GH19 can be either lysozyme or chitinase. Interesting. And then what we did, Adam checked all the genes for peptidase, GH19, GH25. These are all lysozymes. GH3 is a partner of lysozyme to deconstruct further glycans. And then the AMP. So one of them was really highly expressed, okay? So that is consistent with these guys being involved in interacting. We can look at the slides. I can. Yeah. GH19. Are they chitinase or lysosome? They're lysosome. Okay? So they basically degrade the cell wall in a simple basic assay. That is not acceptable, apparently, if you work on bacterial cell wall. But for us, it was a good evidence that it's real. And if you mutate the key residue, it doesn't work. So it's a lysozyme. Now we spoke with Valdemar, the same guy that did the test for the other the peptidase. And we could demonstrate, this is a control, where you use an enzyme from a bacteria, and if you use the Gallinae wild type or T. vaginalis wild type, GH19, they are lysosome. Okay? So now we have a peptidase that targets the peptide of peptidoglycan, and we have lysozymes. So the idea now, we have a complete machinery from a bioinformatic perspective and some experimental data as well, demonstrating that you can deconstruct the peptide bit and you can start to nibble also the glycans. So Trichomonas can target the cell wall and deconstruct it. Okay? That is interesting because that means you can, if you destruct the cell wall of a bacteria, you can eat it easily. It also is the same story as the fungal cell wall. You can trigger inflammation. Now, in addition to this enzyme, we also have a bactericin, an antimicrobial peptide produced by normally bacteria, but Trichomonas, they all have it. T. fetuses as well, by the way. Now, what is this doing normally? This is normally binding, so these are expressed by Lactococcus to kill other Lactococcus. Okay? And how they kill it is by binding to lipid 2. Now, who cares about lipid 2? Lipid 2 is a central intermediate to make the cell wall. This is, for example, what vancomycin binds to. So read your lecture about antibiotics. Vancomycin will kill bacteria by not allowing the bacteria to make the cell wall. That works in the same way. Okay? So we have a very intriguing case of a bacteria, Trichomonas, expressing a bactericin that targets lipid 2. That could be controlling Lactococcus, for example. We have no evidence for that at the moment. Well, we could... yeah, I'll stop that. So we have enzymes that target the cell wall to deconstruct the cell wall, and we have antimicrobial peptide that does not allow the formation of the cell wall. Those two things will be lethal for bacteria. Now, why is the other enzyme GH3 potentially of interest, and why is GH19? Because you can liberate GlUC-NAC. And interestingly, the enzyme I showed you earlier, the first LGT case, is a kinase that phosphorylates GlUC-NAC and GalNAc. And we could show that if you incubate with glucose, you get a certain density. If you have the glucose concentration and add the corresponding half of GlUC-NAC, you get higher growth, like with maltose. So they love it. Okay? So not only do we have deconstruction of cell wall taking place, but we also have the metabolic capability to use a product of that degradation. Okay? And in terms of ATP, you know a lot about ATP production. They make a lot of ATP when you have GlUC-NAC, more than if they have glucose. So Nick did that check. So it's very interesting. And we have the whole set of enzymes that are expressed to, so you can deconstruct cell wall, you can extract GlUC-NAC, and you can metabolize it. Perfect study. And as I mentioned earlier, deconstructing cell wall is a very strong, leads to very strong pro-inflammatory byproducts. So you can imagine in your periodontal pocket, in your urinary tract, when you have two vaginas interact with bacteria, it will liberate these things. The inflammation is going to go to the roof, potentially, because of that. So I think I've stopped that. I've said it 20 million times, and we're all tired, so I'm going to stop here, but I'm not going to show this. This was kind of a summary. Very important, the One Health message. Be very careful who you kiss. Okay? So if your dog or pigeon or wife or husband is treating us, you may ask them maybe to get treated first. So there's clear evidence from the phylogeny I showed you, hopefully I convinced you with that, that there was multiple transfer from pigeons to other hosts. It can be other birds, but it can be mammals, and it can be humans as well. Okay? And then you can find Trichomonas guianae in, you know, what bird is that? Who knows that one? Which one? It's a falcon, yes. Peregrine. And they love eating pigeons. And they get ill sometimes as well because of guianae. Okay? And cats and dogs can have tenax, probably when they eat pigeons. They go that way. Yeah. And then when they kiss us, or we kiss them, I don't like that, but my wife does it. She always does it and to piss me off. We don't have a dog, but we look after dogs. And she does this kind of things and she smiles at me. I hate that stuff. Anyway, just don't do it. And I will finish by acknowledging all the people. Obviously, you guys, I collaborated with you. It was very nice to, and it's still very nice to interact, not only to enjoy the science, but also many other nice things. So thank you for that, and thank you for the invitation again, because it's a wonderful privilege to come here. And the Mediterranean is probably one of the best places on the planet, in many ways, and Sardinia is one of those nice places. And I will finish by saying gracias, gracias, or grazie here. Grazie. Thank you very much. It was a bit long, sorry guys.
Accendiamo qualche luce. Ci sono domande, ragazzi? Meno ragazzi? Has Trichamica ever been found without Trichomonas?
No, we don't see it without Trichomonas.
Nobody tested the
Well, it has not been tested specifically, but in the metagenomics data,
yeah.
you have always support.
Yeah.
I mean, no, you have Trichamica in Italy.
So nobody tested pigeons to look for Trichamica without independently from.
Again, we could do more screening, but the one that being screened, they all have always report.
Like
Exactly.